Hey guys, little video for this week 15 when we talk about patterns of inheritance. And I wanted to start with all about P's, dominance and Punnett square. I want to start with P's first. So this is a beautiful rendition of a pea plant. As you can notice, there are flowers and all things, just roll with it. Uh, the thing I really want us to realize, we know about chromosomes. We know that we carry two chromosomes for each homologous chromosome pairs, as we can see in the lecture notes and in the lecture slides for the karyotype when you see all the chromosomes of human beings. So you have 46 chromosomes and they're all paired up, right? And on each of those chromosomes of a homologous pair, they carry the same genes. So if you take a look at a chromosome, unreplicated, is like that, and there is the homologous chromosome, unreplicated, of the same pair, and each chromosome has the same genes, meaning that if you carry on the gene, on the chromosome here, the gene for eye color, you can carry any allele of the gene here. You can carry the allele for green, blue, brown, purple. You can carry the allele for all of those, but that gene is the location. On the homologous chromosome, you also have an allele at that location. You also carry the gene for eye color here. And so that's why you have two alleles of each genes. So for eye color, you will have two blues, you blue. One blue, one brown, you brown. That's because brown is dominant over blue. Blue is recessive. So a little bit about dominance now. If you to be dominant for an allele means that it's expressed if present all the time. So if any of those is one of a dominant type, it's expressed. To be recessive means you have to have two copies of that gene to be the same recessive allele to see an expression. That's always true, with one exception, the sex chromosomes, and you will learn about that later in this week. Importantly, now that we understand dominance, so we understand that we have homologous chromosomes, pairs of them, 23 of them. Each homologous chromosome in one given pair is carrying the same genes all along. So if here you have genes for eye color, hair color, all of the genes you can think of, they are the same genes here. Maybe not the same alleles of that gene. Remember, it can be a different copies, it can be a different variant. Now that we know all that, when you go to make gametes, you only have one of those two homologous chromosomes going in each gamete because you divide. Meiosis is division, segregation of homologous chromosomes, right? Mitosis is segregation of sister chromatids because you replicate the chromosome and you want to remake cells that are identical to the parent cells. Whereas in meiosis, it's segregation of homologous chromosomes. So you want a packet of chromosome containing one of each pairs on one side for one gamete and the other side for the other gamete, right? Remember, that's the separation of homologous chromosome, which means that gametes have all the genes of the parent cells, but they have only one variant of each of those genes. So when you make those gametes and you go through fertilization, you go back to a pair of homologous chromosomes because a gamete brings one homologous chromosome, the other gametes bring the other one, egg sperm, for example, and you get an individual that has two copies of each genes. Perfect. We get there, we understand all that. Now, when you're a human being and you do fertilization, you make one baby, right? And you produced a ton of sperm cells, a ton of eggs, but only one of them gets food. Now, what's really important for the pea experiment to understand is for us humans, the babies, for peas, it's the little green I want to say pod, but it's not pod, right? Seed. Seed. I think we call that seed. Yes. Sorry for that. So in a pod, it's like here you have 5 babies. Each of those seeds is one, it's equivalent to a human baby. It's just that plants make a ton of seeds, right? So in all the drawings, when we show you one pod for a plant, and we show them all green, for example, if that's the pod that I'm showing you and I tell you all of the seeds made by that plant are green, it's not only that pod, it's all of the pods of that plant. All of the pods, all of the seeds. And it's because that plant only can make green seeds because it only has the dominant allele, right? Whereas when you see any other combination, that tells you a little bit more. That tells you that the gamete can carry either the green dominant or a recessive one, because that particular seed here must have come from the fertilization of two gametes carrying that recessive red color. In my little scheme here, the green is dominant, the red is recessive, right? So that's really the thing to keep in mind. When there's a pod that's shown to you with half green, half red, or whichever combination, that's only one pod. That particular plant, for all what we know, in a given plant, you can have those four pods. Because it's random fertilization, right? A ton of pollen has been made, a ton of eggs have been fertilized, and then you make a ton of pods. So even a plant that we show you a pod that's mixed can make pods that are only green. It's just random chance. But if you take a look at all the pods on one plant, you will see an average of half and half. That's why in the drawings, in the slides, we show you one representative pod of the entire plant. But that plant, it's random what it produces. That's why we get now to the next step of Punnett square, in which we don't represent all potential offspring, but proportions of offspring. So let's work with that. Okay, I dropped the drawings of plants. I'm going to focus on genotypes now. So we cross two pea plants. That's the same slides as the slides. Let's see, it's number 22 in your slide deck, if you want to follow along. So I'm just showing the Punnett square, and I'm going to work with the proportion. The genotype of each parent is big Y, little y. So it's making the, like in that scheme in the slides, we tell you that the yellow is dominant. So if you have the allele capitalized Y, you produce the yellow pigment. If you have the allele non-capitalized Y, you do not produce the yellow pigment. Therefore, you are green. But to express a recessive allele, you need to have two copies of each. So the parent having a genotype of big Y, little y, both of them, they have all of their pods containing seeds that are yellow. All of the pods, everywhere on the plant. So now when we decide which gamete, the Punnett square, it's all about making the column and row headings representing the gametes. A parent that has big Y, little y can make gamete, because meiosis, it's about segregation of homologous chromosomes. That chromosome has a copy big Y, that chromosome has a copy little y. In average, half of the gametes will have a big Y copy, while the other half will have a little y copy. Same thing here. Right? So with that in mind, now we can match them up in those squares. And here we don't say that 4 offsprings are made. Remember that plants is going to make a ton of seeds in a ton of pods. But since in half of the times you have those gametes and half of the time that one or that one, you know in 25% of the seeds, 25% of the seeds will be like that combination, 25 like that, like that, and like that. And that's it. So here you have big Y, big Y, big Y, little y. You always put the dominant allele first, the capitalized allele first. It makes it easier to know the phenotype. And that's the only case when you have two little y's right there. So 25% of the offspring will have green seeds. 25% of the 50% of the offspring will have yellow seeds, but being heterozygous, and 25% of the offspring will be homozygous dominant and have yellow seeds. Making that Punnett square here, it tells you about proportion of those pods, right? So if you want to make a representative pod of a plant, you will make 3 seeds that are yellow and 1 seed that is green. But that particular, those particular offsprings from those parents can totally have pods having only green seeds by random chance. But if you take all the pods and all the seeds of that plant, those will be the proportion you're going to fall on to. That's really important to realize that a Punnett square is proportions. You have to think about it this way. And now think about humans instead of pea plants. You're not going to have a ton of baby in your life. And so that means that that's why we call that a lottery with fecundation in humans. Because when you have one baby, you can fall anywhere, right? 25, 25, 25. It's a quarter chance for each of those. And so you can likely have a recessive. And if that's a disease, you get a disease, right? Whereas for plants, they make so many seeds that, oh yeah, 25% of the seeds have disease and die. Well, you'll still have 75% of plants that don't have that recessive disease and still are alive. In humans, the genetic is way less forgiving because we make less offspring. Okay, so that's a Punnett square. That's how you're supposed to draw them. That's how you're supposed to interpret them. I just wanted to make sure we worked from the basics of what are those? What do they mean? And when we look at the picture of a pea pod with pea seeds in it, it's not the only type that's out there on the planet. That remember, fecundation is random; those are proportions, so it totally means that you can have a pod with only homozygous seeds, a pod with only homozygous recessive seeds. So all greens are all yellow, but when you look at all the pods on these offsprings, that will be the proportion you see. When you start thinking like that, it starts making more sense, in my opinion. Okay, and see you next time.